My name is Elizabeth Jackson, and today we will be discussing cytology, specifically a human cell and its organelles. The first part of the human cell that we will discuss is the plasma membrane, also known as the cell membrane. The plasma membrane is bilayer. It contains the contents of a cell, and it controls what goes in and comes out of cells. It also controls interactions with other cells around it. The side that is exposed to the outside of the cell is known as the extracellular membrane. The side that is exposed to the inside of the cell is known as the intracellular membrane. The plasma membrane is formed mainly from lipids. The majority of these lipids are called phospholipids. Phospholipids are made of a phosphate head, which is hydrophilic and attracted to the intra and extracellular fluid, and two fatty acid tails, which are hydrophobic and float towards each other. This makes them amphiphilic. Phospholipids are not attached to each other in any way, which allows them to float around each other relatively freely. The plasma membrane is made up of other lipids as well, including cholesterol, which we see here in yellow. Cholesterol provides some rigidity around the free-floating phospholipids. Glycolipids are another type of lipid that make up the cell membrane. These are also phospholipids, but they have a carbohydrate chain that extends past the extracellular membrane. These aid in glycocalyx, which helps identify the cell as self. The plasma membrane also contains membrane proteins. Transmembrane proteins extend past the intracellular and extracellular membrane. Peripheral proteins exist on one or the other. Some functions of membrane proteins include communication between cells, enzymatic breakdown, passage of solutes, transportation of molecules, cell identification, and connection to other cells. The cytoskeleton of the cell is on the inside of the intracellular membrane. It is made of proteins and gives the cell structural support and shape. The cytoplasm is everything inside of the cell, which includes the cytosol, which is also known as intracellular fluid or ICF. ICF contains the organelles. Organelles carry out very specific functions within the cell, which we will talk about now. The nucleus of the cell is like the air traffic control center. It is the largest organelle within the cell, and it is central. The nucleus contains our DNA. The nucleus is also bilayer, like the plasma membrane. The layer that covers the nucleus is called the nuclear envelope. The nucleolus is within the nucleus, and it produces ribosomes. We will discuss ribosomes shortly. The rough endoplasmic reticulum, shown here in light blue, is connected to the nuclear envelope and is covered in ribosomes produced by the nucleolus. The rough ER is where peptides and proteins are synthesized from amino acids. The smooth ER, shown here in dark blue, is connected to the rough ER, but does not synthesize peptides and proteins. Rather, the smooth ER helps to detoxify the cytoplasm, which again includes both the cytosol and the organelles. Both the smooth and rough endoplasmic reticulum are made of cisternae, which are these connected channels. The rough ER is covered in ribosomes, like I said earlier. Ribosomes can also be found around the cytosol, not attached to the rough endoplasmic reticulum. Ribosomes read genetic information and produce peptides and proteins based on that information from amino acids. Peroxisomes are formed in the endoplasmic reticulum rather than the nucleolus. They produce hydrogen peroxide within the cell, which helps break down materials that are not wanted or needed. The Golgi complex finishes peptide and protein production from the ribosomes. They also create carbohydrates. The Golgi complex, like the endoplasmic reticulum, is made of cisternae, which are flat sacs closely packed together. Completed peptides and proteins are placed into Golgi vesicles, which bubble off of the Golgi complex. Golgi vesicles transport the completed peptides and proteins within the cell cytoplasm, which is called endocytosis. They sometimes transport peptides and proteins through and out of the cell membrane, which is called exocytosis. Lysosomes are a type of Golgi vesicle. They move around the cytoplasm and break down particles and even organelles. Some functions include breaking down bacteria and releasing stored glucose into the blood, depending on which type of specialized cell they are functioning in. The mitochondria are like the oil factory. They create energy for cellular function. This energy is in the form of ATP. ATP is created from things like sugars, proteins, and fats. The mitochondria themselves are oval-shaped. On their inside are folds called cristae. Cells may have more or less mitochondria depending on how much energy they use. Finally, we have our centrioles. They are made up of sets of 3 protein microtubules. There are 9 sets total arranged in a cylinder. The centrioles are used in cell division. Their function is to ensure that cells divide correctly by moving around the parts within the cytoplasm.